Good morning, my name is Fernando Torres Andón, and first I want to thank you very much for this invitation and this opportunity to participate in this workshop. I will talk about the therapeutic manipulation of macrophages for the treatment of cancer using nanoparticles. I have started to work on this idea 6 years ago in the frame of a collaboration between the lab of Professora María José Alonso at the University of Santiago de Compostela in Spain and the lab of Professora Paola Alavena at Istituto Clinico Humanitas in Milan, Italy. Investigations at Humanitas Institute and other many institutes around the world have demonstrated that most of solid tumors present a major population of macrophages, which in some cases can represent up to 50% of the tumor mass. These have been denominated tumor-associated macrophages. On the right, we can see some examples from our lab on tumor tissues from immunocompetent murine models presenting a high infiltration of macrophages. Here we can see a lung tumor and the macrophages in brown and a fibrosarcoma tumor. On the left we can see samples from a human patient with lung cancer, and we can see by immunofluorescence the macrophages in the lung and also by immunohistochemistry the macrophages in the tumor lung tissue. Importantly, it was observed that these macrophages in tumors present pro-tumoral functions, and they are key responsible of the immunosuppressive tumor microenvironment. These tumor-associated macrophages present a series of pro-tumoral functions. They promote proliferation and survival of cancer cells, angiogenesis, invasion and metastasis, genomic instability, tissue remodeling, and fibrosis. In addition, these tumor-associated macrophages are responsible of suppression of adaptive immunity and resistance to antitumoral therapies. However, the good news is that these tumor-associated macrophages can be reduced into antitumor macrophages. Macrophages are very plastic cells, and they can differentiate into a wide variety of functions and phenotypes. This heterogeneity of phenotypes and functions can be theoretically divided into two extremes, which have been denominated M1 and M2. In cancer, tumor-associated macrophages present an M2-like phenotype with pro-tumoral functions, and on the other extreme, the M1-like macrophages present antitumoral functions with ability to kill tumor cells, inhibit angiogenesis, and promote adaptive immune responses. So we have decided to work on the development of nanomedicines to eradicate tumor-associated macrophages towards M1-like antitumor effectors. With a lot of interest, these antitumor macrophages result very interesting for combination therapies with radiotherapy, immune checkpoint inhibitors or CAR T cells, immunogenic cell death activators, or targeted therapies. Several drugs have been investigated for their ability to reprogram macrophages towards M1-like antitumor effectors. We have decided to focus our attention on the activation of intracellular Toll-Like-Receptors by poly-IC and resiquimod. Poly-IC is agonist of TLR3 receptor, which activates the TIRF-IRF pathway, resulting in the production of type 1 interferon responses, co-stimulatory molecules, and secretion of T-cell attracting chemokines such as CXCL10. Resiquimod is agonist of TLR7 and TLR8 receptors that activates NF-kappa beta pathways and MAP kinases pathways, resulting in the activation of survival signals and production of pro-inflammatory cytokines. Also, there is an indirect activation of TLR3 of NF-kappa beta and also indirect activation of TLR7 of IRF7, producing similar effects when it comes to macrophage activation towards M1 antitumoral phenotype, and giving also some possibilities for synergistic effect of these drugs. TLR agonists have been used as adjuvants for the treatment of cancer for several years. There are several clinical trials ongoing, and The major problems for the administration of these drugs are related to the poor stability and systemic toxicity and difficulties to reach macrophages in the tumor. These are the aspects that we aim to improve with the development of nanomedicines. So now the question is, how to improve the delivery of these TLR agonists towards macrophages using nanoparticles? Okay, we know that when we administer nanoparticles into our body through different routes, most of the nanoparticles are recognized by innate immune cells, and this interaction results in the triggering of immunostimulation or immunosuppression, the biodegradation of the nanomaterials, or adaptive immune responses. In 2014, Weisleder et al. Have showed in this review the preferential uptake of nanoparticles by macrophages or dendritic cells versus other cancer cells or immune cells. The horizontal bars represent variability between tissues, and below these balls represent the amount of nanoparticles accumulated in different tissues along the body after intravenous administration, which is largely due to the size of the nanoparticle. For example, bigger nanoparticles end up in lung or liver, while small nanoparticles smaller than 10 nanometers are eliminated through the kidney in urine. So we aim to develop nanoparticles with a size between 10 and 200 nanometers for the treatment of cancer. Later, in 2016, Wilhelm et al. From Toronto published this controversial meta-analysis showing that most of the nanomedicines which have been developed till now for the treatment of cancer are only able to increase the amount of drug reaching the tumor in a small percentage of 0.7%. In a subsequent study in 2018, they have demonstrated that functionalization of nanoparticles with ligands to reach tumor cells is not able to improve this 0.7, and a very small percentage of drugs reach the cancer cells. These results are controversial, and many other research groups in the last years have reported of specific types of nanoparticles improving this delivery. However, what is commonly accepted in the community is that macrophages are more efficient than cancer cells taking up the nanoparticles. When the nanoparticles reach the tumors, in the tumor vessels, the nanoparticles extravasate and most of them accumulate in the macrophages in the vicinity of the tumor vessels. In this study, they show this accumulation in macrophages for both nanoparticles for active and passive targeting. If we look at the nanomedicines for the treatment of cancer which are used in the clinic, we can already find some examples. In this review from Mirkin et al. In 2015, we can see that 12 nanomedicines have been already approved by the FDA. The most well-known are Doxil, which is liposomal doxorubicin, or Abraxane, which is albumin-bound paclitaxel. In this process of designing a nanomedicine, the ideal nanomedicines for its intravenous administration, upon injection in the intravenously, the nanomedicine should be able to avoid the interaction with serum proteins. This nanomedicine must be able to increase the plasma concentration of the therapeutic drug or increase the time in circulation, and then achieve a high accumulation in the tumor. Once the nanomedicine reaches the tumor, it must be able to penetrate, to extravasate from the vessel, reach the target cell, in this case the cancer cell, and then release the drug at the appropriate intracellular compartment where the drug has to do the effect. So the drug must be released at appropriate time, and the drug must be still active. So this is what we have started to do. On one hand, we have prepared a series of polymeric nanoparticles, nanocapsules using different polymers, inulin, dextran sulfate, polysialic acid, hyaluronic acid, polyarginine, protamine, ketoxan, and loaded with resikimod. These nanoparticles present different surface charge and different size, but with ideal size between 70 and 150 nanometers, and different surface charge from positive 50 towards minus 30. On the other hand, we have developed poly-IC nanocomplexes using polyarginine or C12-octarginine polymers, which have been then coated with a protective layer of hyaluronic acid or PGA-PEG. Then we have started to work on the in vitro evaluation of the poly-IC nanocomplexes and the resikimod nanocapsules. Here I will present only the results for the poly-IC nanocomplexes as an example. We isolated monocytes from healthy blood donors treated with MCSF to differentiate them towards macrophages. Then we treat the macrophages with the poly-IC nanocomplexes, and here we evaluate the toxicity of the nanocomplexes to the macrophages. We treat the macrophages with different concentrations of poly-IC, and we observe that those response toxicity, and we can select a concentration of 5 micrograms milliliter of poly-IC for all the nanocomplexes, which is non-toxic to the macrophages. Then, using the similar setting, we evaluate the secretion of the cytokines of macrophages treated with the nanoparticles. Here we present the results for CXCL10, which is a cytokine secreted by the macrophages to attract T cells to the tumor to fight against cancer cells. Here we observe in the results a higher secretion of the CXCL10 by the nanocomplexes, by the macrophages treated with the nanocomplexes versus the macrophages treated with free poly-IC. Here another functional assay that we have optimized in our lab. Here, in this case, we isolate monocytes from human donors, healthy donors. We treat them with MCSF, differentiate towards macrophages, and treat the macrophages with the nanocapsules. Then, these macrophages are incubated with tumor cells, and we evaluate the ability of the macrophages to kill the cancer cells. At the end of the experiment, we collect the cancer cells and the macrophages, and by FACS, we are able to quantify each population. Here we have one example, and the response to different treatments. Here we quantify the cytotoxicity of the macrophages towards the cancer cells, and we observe a similar ability of macrophages treated with free poly-IC and the macrophages treated with the nanocomplexes to kill cancer cells, which is very important. That is very similar to the prototypical M1-like macrophages versus the non-treated macrophages, which are not able to kill the cancer cells. In addition, we have evaluated the uptake of the poly-IC nanocomplexes by macrophages. For this, we have used poly-IC labeled with rhodamine, which is a fluorescence molecule, which we can quantify the amount of fluorescent signal inside the macrophages. So, when we treat the macrophages with the poly-IC free or the different nanocomplexes, we can see an increase for the polyarginine poly-IC nanocomplexes versus the free poly-IC, which is prevented in the case of the coating with PGA-PEG and partially prevented by the coating with hyaluronic acid. Then, by confocal macroscopy, we evaluate the localization of the nanocomplexes inside the macrophages. Here we have stained we can observe in these pictures the poly-IC in red, the lysosomes in green, and the nucleus in blue. The overlapping of the signal from the poly-IC nanocomplexes with the lysosomes makes this yellow signal, which is very clear, showing the colocalization of the poly-IC nanocomplexes with the lysosomes in macrophages. where they should exert the therapeutic activity on the TLR3 receptor. Finally, I will present some examples of in vivo experiments. Here I'm not presenting nanoparticles because we have still not published our results. So just an example and the methodology how to do these experiments. So we have used subcutaneous injection of cancer cells in immunocompetent mice, C57. So we inject these cells and we wait 9 days to the growth of the tumor cells, and then we injected intratumoral these treatments, poly-IC or Resikimod, for 5 injections. Then we observe that the intratumoral injections of poly-IC and Resikimod are able to prevent the tumor growth of these tumors in vivo versus the control. Here we can see at the end of the experiment the results for the poly-IC and for the Resikimod, and now the question is, we will be able to improve this therapeutic activity with the use of nanoparticles loaded with these drugs to target macrophages in the tumor? Now I will present also our work on the optimization of immunocompetent orthotopic in vivo lung tumor models. We inject the cancer cells intravenously or orthotopically in the lungs of the mice, and after a therapeutic window of 3-4 weeks, we sacrifice the mice and we collect the lungs. One of the lung is dissociated we prepare a suspension of the cells. This suspension of cells can be used for a colony assay and also for FACS analysis to evaluate the immune infiltration, in this case with a particular focus on M1, M2 macrophage polarization. The other lung is prepared for immunohistochemistry to evaluate the infiltration of immune cells, in this example macrophages, and also for hematoxylinious zinc staining to quantify the tumor load. Here we present a correlation of the number of colonies and the tumor area evaluated by hematoxylinious zinc, presenting a very good correlation. So as a whole, we think that this protocol allows us to quantify the tumor load and also characterize the immune infiltration in these tumors. Here one example of mice with tumors in the lung treated with poly-IC. We can see for the control a high tumor load, which is prevented by the poly-IC. By the colony assay, we can see some examples of hematoxylinious zinc staining, also showing the same result, and also a good correlation with the tumor, with the weight of the lungs. In the same experiment, we can see by immunohistochemistry the infiltration of CD3 T cells in the tumor, which is increasing by poly-IC, and also we can see by flow cytometry an increase in the population of myeloid dendritic cells, also NK cells and T cells. As a whole, I hope to give you these take-home messages. Reprogramming tumor-associated macrophages towards M1 antitumoral effectors is very important to fight against immunosuppression in tumors and to reactivate the immune system to fight cancer. The optimization of immunocompetent in vitro and in vivo models results crucial for the evaluation of TAM-targeted therapies. Polymeric nanostructures offer a wide variety of systems which interest the promising properties to load and improve the delivery of combinations of immunostimulant drugs towards TAMs to fight against cancer. Finally, I want to acknowledge all the funding which allows us the performance of this work, the funding from the EuroNanomed project to Intratarget, also Worldwide Cancer Research Association, also Marie Curie funding from the Horizon 2020, Xunta de Galicia, the regional government, and also the Spanish Association Against Cancer. Also, all the people involved in this work, from the lab of Professora Paola Lavena in Italy and from the lab of Professora Maria Jose Alonso in Spain. Thank you very much for your attention and have a nice day.